Hi everyone, this is Dr. Saud Malik here. Welcome you all the future doctors and all the aspirants. This time I'm here to discuss an important topic, which is from an important lesson. That one is the inheritance phenomena, or the molecules which are responsible for the inheritance phenomena. And this DNA structure is one of the very important part of it, and it will help you a lot when you will prepare that. Surely that the weightage of this particular topic is very high in the examination, and on top of that, that will open up all those windows which are very necessary to know more about the cell biology or the genetics. So let's move ahead, and first we will try to understand what is that. As you know all, we are always looking like very similar to our parents, right? Most of the time we are looking like some people amongst us are having more feature of like their mother, more features like father, and even in the other animals, if you will observe that, you will see that they are also looking like very much similar. On most of the aspects, they are quite similar to their parents. But the question is that, who is taking care of this thing? Why this thing is happening in case of the life? Why all of us are looking more like our parents? Or why other animals are looking like their parents? Answer is very simple. Definitely something is there which is taking care of it. And my dear students, that is DNA. A very mysterious, a very magical molecule which is creating a very fascinating biological world. And surely today we will try to decode each and everything about it. First of all, I would like to know that you must be aware of that. What is the full form of that? Yes, deoxyribonucleic acids. That is the very correct full form of it. And let's open up each and every aspect of this knowledge for you all. So let's move ahead, and first we will take that deoxyribonucleic acid, which is a necessary part of each and every cell. Obviously, because each and every cell needs a kind of nice control on it. During the process of the reproduction, that is very important to ensure that a living will give birth to the like ones. And that is the reason all prokaryotic cells and eukaryotic cells are well equipped with it. That is very important. And from the chemical point of view, if you will talk about, like, there are multiple kind of the molecules in the cell. Out of them, this molecule belongs to nucleic acid, a very special class of the molecules. So nucleic acid is the category from which this molecule is coming. And as I was telling you, this molecule is definitely transmitting the hereditary material or the genetic information or instructions from parents to the offspring. Definitely, when you will go through this understanding or this study, you will come to know viruses, most of the viruses do not have that kind of DNA. So that is actually an exception in case of some viruses. They may have RNA as their genetic material. When you will compare both of these molecules, so surely you will come to know there are a lot of similarities in between these things. So RNA is a kind of the other option in case of the viruses, and it is used there as the genetic material. While others have DNA as the genetic material, like we all, our genetic material is definitely DNA. The molecular structure of DNA is basically double-stranded structure. If you will see that what kind of that double-stranded, definitely we will discuss that. we will reach to the level of that final structure of this molecule, then we can understand it. But still here, I would like to give you a meaning of that double strand. Double strand means when you will see that this is a single strand I'm drawing over here. And one more strand, if I can connect through it, that will look like that. And these two strands, this one is the one strand, any strand you can say that it is one of these strand of it, and this one is the other strand. So both strands are needs to be there together, and which are nicely connected with some bases over here. We will do a detailed study of it, don't worry about that. This is just to give you an idea about that what is the double strand structure over here. So that is a double stranded molecule, two strands, and these two strands wrap around one another and form a shape which is known as double helix. My dear student, this is very important word. DNA is a double helical structure. One helix means that one strand is over here and second is there. That is the single helix. Double helix means that two things are coming together. That is known as the double helical structure of DNA. We will discuss that in detail. Don't worry about that. And alternating phosphate and sugar groups are there. Apart from being responsible for the inheritance of genetic information, DNA not only playing a role of carrying the genetic information from one generation to another. Beside that, dear students, it is doing something else, and that is related to the protein formation, that is related to building new proteins in all over the cell as per the requirement of the cell, as per the demand of the cell. So that is also one of the most important part of the DNA molecule which is doing. All right. Now if we move together, some historical knowledge, we should collect it. If you will try to collect that historical information, remember that it is a vast information. So here we are not doing that in the details because we are taking it just from the information point of view. DNA was identified in 1869 by Miescher, Friedrich Miescher, a very famous scientist. Due to technical limitation, structure was unknown till decades. That wasn't clear how that thing is arranged there inside the cell and what will be the appearance of it, what will be the structure of it. Because as you can understand that 1869, a very, very early era for the science, biological sciences. So not that much of information was available to understand that. But gradually when people came to know, when scientists came to know about the nice microscopy, lot of arrangement in the laboratory, so they came to know that DNA in detail is very much like which we know in these days. So double helical model of the DNA first propounded in the 1953 by James Watson and Crick. Remember that. That name is very important. Watson and Crick model is still, we are following that. That Watson and Crick model was not only qualitatively as well as quantitatively perfect. So they explained it in the detail in a research paper, and we are following that. What they did, actually, they were using a very special technology which was recently launched in the 50s. And that was X-ray diffraction data produced by the Maurice Watkins and Rosalind Franklin. So they were using that data, they were using that information, and finally they were able to give us a very detailed model of the DNA structure. All right, hope the things are going on very well. Now let's come to the most important part, which is known as the structure. As I was talking about in the initial stage, DNA is a polynucleotide. Remember that polynucleotide means it is made of many nucleotides. So it means that DNA must be made of nucleotide. Yes. So the unit of the DNA structure is nucleotide. So when many nucleotides will come together, that will create a polynucleotide. And they form just like a chain. You must be having a kind of the mala, garland, or any other kind of this thing at your home, in which the beads are connected one by one, one by one. Similarly, all these nucleotides are connected well in the form of the chain, and that gives us a very, very important structure. So what are the components of this nucleotide? If you will see that nucleotide is basically made of three things. The first thing is the carbohydrate molecule, that is the sugar. It is basically a pentose sugar. That is a pentose sugar. Pentose sugar means that 5 carbons are in this structure. That is the reason it is known as the pentose sugar. And a nitrogenous base, this is the second part of this structure, and a phosphate group. So these three things, when nicely compiled, when nicely arranged together, that will make a kind of the nucleotide. So this is the basic structure of the nucleotide. So how this thing is happening? Nucleotide is not directly made. First of all, that nucleotide is formed by the sugar and nitrogenous base only. Can you see that? Sugar and nitrogenous base. Only these two things are there. And these two things are giving a nucleoside. So first nucleoside will form. Then nucleoside, when connected well with the phosphate group, that will give you a nucleotide. So one by one, these things are nicely compiled. So nucleotide, when arranged by the process of the polymerization, that is the name of a chemical reaction in which many monomers connect together to create a big molecule, a big chain, which is called polymer. So in other words, that polymerization, who is going to be the monomer of this process? Yes, nucleotides are going to be the monomers of it, and that will give you a long polynucleotide chain, which is called DNA. Here, a very easy illustration is given over here. You can see that a pentose sugar molecule, that is the sugar molecule which is present over here. That pentose sugar molecule, it's here, and that sugar molecule is connected with the phosphate group and the nitrogenous base. So my question is that where is the nucleoside in it? This is the nucleotide. If you will see that sugar and nitrogenous base. So this much area only, I'm talking about this much area. Yeah, that is going to be a nucleoside. And when this nucleoside will connect with the phosphate group, that will give you a nucleotide. And how this thing is happening here, we have a small illustration to show you that how this bonding is taking place over here. You can see that the pentose sugar in which 5 carbons are placed. So this structure is telling you that oxygen atom is over here. So this oxygen atom is giving you an idea about it. And all these points are covering carbons over here. All right. So if I'll say that this is the first carbon, this is the second carbon, that one is going to be the third carbon, fourth carbon, and fifth carbon is visible over here. Can you see that? In this way, these 5 carbons are in it. It is the structure of a pentose sugar. That sugar is known as the ribose sugar. Remember that. This is the ribose sugar. So that ribose is connected to this much of the part, that is the phosphate group, and here is the nitrogenous base. This bonding we will discuss in detail, what kind of bond are they, and how these things get nicely connected in this way. That chemistry will also come into this discussion, definitely. Sugar component of DNA, one by one we are discussing all the component of it. As I was talking about that sugar, phosphate group and a nitrogenous base. These three components need to be discussed separately. So let's come to the first one, that is the sugar component of DNA. So the carbohydrate or the sugar part of the nucleic acid, basically a pentose sugar ring. Okay, as I was telling you, 5 carbons atoms, and they arrange in a cyclic manner from 1 to 5. See the difference in both these kind of the sugar, you can understand it well. What is the basic difference of them? This basic sugar is the ribose sugar, and this one is the deoxyribose sugar. That is going to be the part of the DNA, deoxyribose sugar, and this one is going to be the part of the RNA. So both these things are going to be different just because of this contain. So what is basically the difference in them? If you will see that, look at this. The 2 prime, the second number of the carbon in this ring. If you can see that, here is the OH over here. And in case of the deoxyribose, that hydroxyl group will lose the oxygen of it, it will be H. So that is the basic difference. You can understand it. Just because of that loss of that oxygen atom from here, it is deoxyribose structure, and that one is the ribose structure. Okay, so this difference is very, very important to understand, the difference in between the RNA and DNA. That is the root cause by which these two molecules are quite different, and it will change the whole chemistry of both of them. All right, let's move ahead and try to find out what is basically the links in between them. So let's come in a nucleic acid, these sugar parts are linked with the phosphodiester linkage. Remember that phosphodiester linkage or phosphodiester bond. That is very important. This is basically for the sugar to connect with the C5 over here. Can you see that? As I was talking about it, if you will see that, you can understand it better. In the case of the carbon number, this is the first carbon, this one is the second, this is the third, that is the fourth, and fifth carbon is here. So what is happening? The C5 of the 1-deoxyribose or the ribose in case of the RNA, and that will connect to the hydroxyl group. All right, look at this. This is quite visible over here. Okay, so C3 of the other one by the phosphate group, and the other one, look at this, this is the other one, that one is the other one, this one is the first one, because we are going to that side. Flow is here. So you can see that 1, 2, 3, 4, and this is the fifth. So look at this. That is the third, and this is the fifth. So in between them, phosphate group present on the C5 form a phosphodiester bond with the hydroxyl group on the C3. So C5 and C3 means these two things are going to be the part of this bonding, and phosphate is connected in between them just like a clench. In this way, that creation of the bond is known as the phosphodiester bond. So sugar to sugar in a molecule is connected with the help of the phosphate, which is responsible thing to give contact between two sugar molecule. So phosphodiester bond are very strong and they are covalent bonds. Means they do not break quite easily. When you will give it heat or when you will give it high temperature, it can tolerate well. That is giving a nice stability to the molecule of the DNA. All other sugars are not suitable to fit here. This is always a question. Why the other sugars cannot be the part of it? Why hexose cannot be there? Why haptose cannot be there? Why other cannot be there? Very simple. All of the sugar are not suitable for this structure because of steric hindrance. You must be aware of that effect in the chemistry. That steric hindrance is responsible to create a kind of instability in the bonding structure. That will not allow the things to be together. So it is happening, and various groups present on it, they are responsible for that steric hindrance. Ultimately, other sugars, if you want to accommodate at the place of that sugar, no way. There is no solution at all. So that's all is about the important part of it, that is the sugar. Now let's move to the other part of it, nitrogenous base. As you know, students, nitrogenous base is a really very important part of it, which is giving a very special character. So nitrogenous base were identified in 1880 by Emil Fischer. These are basically simple kind of organic compounds, and they are heterocyclic in nature and planar in form. These are the basic chemical kind of the characters. On the basis of the structure, nitrogenous bases you can group into two parts. The first group is of purines and the other are the pyrimidines. These two are the groups. So we will discuss them one by one. So first I'm taking care of the purines for you. Let me tell you that in the size, these purines are bigger because they have two cycles in them. Can you see that? Two purines I'm taking here like adenine and guanine. You must be aware of that. These two things are looking like made of two cycles over here. Can you see that? This is the first cycle, this is the second cycle. Same here, this is the first cycle and second cycle. So purines are double ringed structure. Very simple. That is the reason these molecules bigger than the pyrimidines. Remember that. And if you will number them, you need to number them in the anti-clockwise direction. The major purine that you may have heard of earlier are adenine and guanine. These two are very popular because they are going to be the part of the DNA and RNA. But beside that, there are so many other kind of the purines are also available. Why we are not talking about them? Because irrelevant knowledge, not useful for us because we are focusing towards the DNA. So why should we go for the other class of those things or other categories of those things? That is the reason. Look at the structure for both of them. You will see this first ring is connected to the second ring in this way. That is basically a structure which is related to the chemistry. When you will do detailed study in chemistry about the biomolecules, you must go for the details about these kind of bondings and everything in it. Here we are covering that from the biological perspectives, so not going up to this depth. Surely in any lecture when I will cover those biomolecules from the chemistry point of view, I'll take care of that thing also. So don't miss those videos also. Let's move ahead and try to find out something about the pyrimidines. As I was talking about, they are made of the single ring. You will notice that purines are composed of two ring and pyrimidines are made of single ring. They are cytosine, it is abbreviated as C. This is uracil, abbreviated as U, and this is thymine, this is abbreviated as T. These three kind of things are available from the RNA and DNA point of view. Pyrimidines hence contain single ring and are numbered clockwise. Means you need to give them number in this way, clockwise. In case of purines, that was anti-clockwise. Okay? So pyrimidine component of RNA is uracil, is the counterpart of thymine. Definitely you are getting this. This is the basic difference. So pyrimidine in RNA is uracil. This thing is going to be only in the RNA. And this thing is only going to be in DNA. So that is the difference in between the RNA and DNA. These two things are different irrespective of the U and T. you should focus towards their other structures also. Okay? Remember that in our entire DNA, nitrogenous bases are always present in the syn form. That is very important to understand what does it mean. It is very simple kind of the understanding. I would like to take you on a screen, just want to show you that what is the meaning of that syn and anti form. These two forms are very important to understand how these two strands will come in contact. If you will see that nitrogenous base is linked to the sugar molecule and have the capacity to be present in two forms because of the rotation of the bond joining the sugar molecule. Can you see that? That rotating axis is visible over here? And just understand it, this molecule is like that and this other part of it is looking like that. And if you will rotate it, it can go this way. Just see that. Just see my hand. If I'm turning it inside, if I'm turning it that side, and in this case, I'm turning it outside. So that place for the attachment, this place is for the attachment to be with the other strand, that is going to be possible. In this way, that is also possible. So I mean to say, this arrangement is giving a chance to connect these bases of both strands together. So remember that this thing is very simple from the geometrical point of view. If you will keep that thing in your mind, you can understand it. If I'll put two strands together and if I want to connect them in this way, so obviously I need to provide a nice attachment over here. If something which is going to be connected within this area is diverted outside or deviated outside, obviously it won't come inside. That is very important to understand. So remember that syn and anti forms are like that. So what we were talking about? We were talking about that syn form is only the way by which they can connect well, and that is here. That is the syn form. I hope this thing is clear to you. All right, so let's move to the first formation which will take place. That is the formation of the nucleoside. One by one, all things will come together. Now, this is the time to compile them, to arrange them, just like a sandwich. If you are preparing a sandwich at your home, you must be having all the content over there. Your bread slices are ready, your vegetable slices are ready, your cheese is ready, your butter is ready, spice is ready. But now the time to arrange them to create a nice delicious sandwich. Similarly, if you want to create DNA, so start arranging them, and the first arrangement is the nucleoside. So nucleosides are glycosylamine compound that form the structural foundation of nucleic acid. They are basically one of the nitrogenous base. It may be purine or pyrimidine, doesn't matter. Both of them will be involved in it. And by the covalent bonding, remember this part, by the covalent link to a 5-carbon sugar, it may be ribose or deoxyribose, they will connect it. And phosphate group is not there. Once the phosphate group will come into it, that will be nucleotide. So this time we are talking about the nucleoside. So they serve critical roles in cellular metabolism, energy transport, and many therapeutic purposes are solved with it. Many therapies now develop well to treat the patients for the problems like cancers or any genetic troubles. So many activities in the research and development are happening just because of these things. So nucleosides components are basically two. The first, a nitrogenous base, and second is a 5-carbon sugar, which is pentose. As I told you that pentose is of two kinds. Ribose is going to be in case of the RNA, and deoxyribose is going to be in case of the DNA. All right. That is important. So how many kinds of these nucleosides are going to be possible? Very simple. The first is made of adenine and the ribose. So it is going to be adenosine. Similarly, guanosine, that is guanine and ribose, cytidine and the uridine. And last one is the thymidine. So these are the 5 categories because all bases are going to be 5, right? So this is the way the all ribose kind are made of. But in case of the thymine, it is going to be deoxyribose because that is going to be the part of the DNA. That's why we are putting it separate. Okay. So these all nucleosides are available. Once these nucleosides will form, now the final structure of the nucleotide, which is the monomer of this structure, will form. How this thing is formed, as you can see there, I explained you well, this is the pentose sugar molecule. And through the pentose sugar, this phosphate group and nitrogenous base are connected. And in this way, that nucleotides will form. So DNA nucleotide is basically contain deoxyribose sugar. So when you will see that, the number of the phosphate group will give it different name. For example, deoxyadenosine monophosphate. What does it mean? That means that deoxyadenosine, which is a nucleoside, this one, and a single phosphate will be connected with it. This is the single phosphate. Means monophosphate, that is known as the high energy bond. They are known as the high energy bond. If the number of these bonds will increase in it, if I'll increase the bond, like if I'm putting one more bond with the phosphate, that will be diphosphate. If I'll put one more over here, that will be triphosphate. Alright? So you must be aware of that structure AMP. Alright? If that 2-phosphate will come over there with these 2 high energy bond, that will be ADP. And if the third one will come over here, that will be ATP. So all of them can give you 3 structures. In case of the DNA, they are known as the DAMP, means deoxyadenosine monophosphate, which is the part of the DNA. Similarly, deoxyguanosine monophosphate and deoxycytidine monophosphate and deoxythymidine monophosphate. Alright, monophosphate means single phosphate, as I explained you over here. Di means 2, tri means 3. That structure is very much relevant to their energy level. When you will discuss the protein synthesis, when you will talk about the replication of the DNA, then these things are going to be important for you. You will understand it well over there too. Alright. Now let's come to the other part of it. That is the primary structure. Now what we are going to discuss in the primary structure and secondary structure of the DNA. Obviously, every molecule has some complex arrangement of the things. So first, these things will get together to create a simplest form. Then it will be more complicated form. For example, I want to create a rope. So first I would like to create some those very fine kind of the initial ropes. Then I'll take them together to create a nice thick kind of the rope. So the previous one was the primary structure, which will be changed into the secondary structure, which is the more complex structure of a molecule. So same in case of the DNA, the primary structure of DNA refer to the phosphodiester bond backbone. This thing we are talking about. That thing we are talking about. This phosphodiester backbone. I hope you can understand this. This backbone is the backbone of the DNA. And this is the arrangement. You can understand it when you will connect that knowledge which I gave you in the initial part of this lecture, where these phosphate things are picking up one by one sugars, one from top, one from down. Look at this. This one, that one. This one, that one. This single phosphate, I'm talking about here, is taking care of these 2 sugars to be together. is a phosphodiester bond. We were talking about it. And when these phosphodiester bond will come one by one in a sequential manner, that will give you a primary backbone of the DNA. It is very much like a ladder, this ladder part. Okay, these are two primary things. Once these things will come together in this way, that will be a secondary structure. When you will see that definitely nucleic acid strand nucleotides are linked by phosphodiester bond, and a single nucleic acid strand formed by phosphodiester bond has two termini, namely 5' and the 3'. Look at this. When you will see that this end is called 5' phosphate end, and this end is known as the 3' phosphate end. The question is that why these names are coming? It is related to the free kind of carbon atom in the sugar molecule. If you will see this particular sugar molecule, let me point it out to make it clear for you. When you will see that, you will see this is the first carbon, this is the second, this is the third. All right? This one is fourth, and this one is the fifth. Let's talk about this sugar. That is also 1, this is 2, this is 3, this is 4, and that one is 5. So look at this. In this case, which carbon is going to be free? If I am taking it as the end of this chain, obviously this fifth carbon is going to be a free carbon. That is the reason it is known as the 5' end. We write it in this way. Similarly, if I take it as the third one, it is going to be a free one. That's why this end is known as 3' hydroxyl end. Why the 5' end is known as the phosphate end? Because at the fifth, only phosphate is visible. This one. Got it? But here, who is going to be available? Look at this. Only hydroxyl group is going to be available. That is the reason it is known as the 3-hydroxyl end. So both of these things are here. One negative charge per residue is coming just because of each phosphate group keep giving one negative charge on this structure. So negatively charged whole phosphate backbone will create in this way. One in this side and second is going to be the other strand. Now think about it. Is it primary? Yeah, that is the primary structure. Now let's move to the more complex form of it. That is the secondary structure of DNA. Look at this. This is the one primary over here. This one is the first primary structure. All right. And this one is the other strand as the primary structure. When these two structures are coming in contact to create a ladder-like structure, that is called the secondary structure. So this is the sugar phosphate backbone, and look at this. This backbone is having 5' over here and 3' over here. The other one is the 3' over there and 5' is there. Means they are opposite. That is the reason these two strands are said to be anti-parallelly arranged. They are known as anti-parallelly arranged. Anti-parallel arrangement. This is known as. Now these things need to be connected to create these kind of connections. Look at this. This connection is very interesting. As you can see here, A always have affinity with the T, and they are nicely arranged with the two hydrogen bonds. Can you see that? This one is quite clear over here. You will see that two hydrogen bonds are visible over here. And in case of G and C, guanine or cytosine, they are basically 3 hydrogen bond. So the connection is like that. A will always go with the double bonding T and G will go with triple bonding C. In this way, all things are get arranged and that is known as the secondary structure of the DNA. I hope this all is clear to you. If you have any query and any question, please keep those questions for the next lecture. In the next lecture, first we will discuss whatever we are discussing over here. That will be a quick recap, and after that we will cover the next part of it. Till then, keep learning. Bye-bye.